I'm Trent Hayden, and I'm a postdoctoral associate at CGSB in the New York University, Abu Dhabi, and I'm studying the high thermal tolerance adaptation of Arabian Gulf corals. The coral reefs in this region are extremely unique compared to other coral reefs. The Arabian Gulf reef water temperatures here are the hottest in the world and frequently hit 35 degrees or higher during summer. Coral reefs generally shouldn't exist in such high temperatures, but the corals in Abu Dhabi have genetically adapted to survive in these environments. Despite the high temperature tolerance of corals in this region, Abu Dhabi's coral reefs are currently facing threats from climate change and other local stresses like many other coral reefs worldwide. It is crucial to protect and conserve these reefs to preserve the biodiversity they support, sustain the ecosystem services, and ensure the long-term well-being of both marine life and local communities of Abu Dhabi. The local communities here rely on the healthy functioning of these reefs as an important source of both food and income. Therefore, it is no surprise that we're interested in understanding the fundamental mechanisms of the high thermal tolerance of these corals. The future persistence of coral populations globally depends on their ability to increase their thermal limits at a pace in line with the rapid warming of our seas from the effects of climate change. One area of particular interest is understanding the role that the coral microbiome may play in coral adaptation and response to thermal stress. Similar to the human microbiome, corals associate with a diverse group of beneficial bacteria, which we think are critical to the success of these corals within this hostile environment. We're currently working on isolating as many individual bacteria associated with corals as possible, so we can try to figure out which ones in particular may enhance a coral's ability to survive the high temperatures experienced by Arabian Gulf corals. One of the advantages of working with microbes is their short generational times. Microbes can evolve adaptive functions much quicker than the coral can. So by exposing individual microbes to different environmental stresses, we can start to understand some of the compounds they produce and metabolize, which promote coral health and resilience to stresses. By identifying and defining the specific roles of beneficial microbes associated with corals that we have in isolation, we hope to generate a probiotic treatment for corals that may reduce the negative impacts of thermal stress.